back to biotech. Then here we are going to see the first chapter, full chapter we are going to see with the most important points with quick revision. చాలా తొందరగా ఎలా క్విక్ రివిజన్ చేయాలో అలా ఈచ్ అండ్ ఎవ్రీ పాయింట్ వితౌట్ మిస్సింగ్ ఎనీ వన్ బిట్ maximum everything I have been covered, so that please follow me. Those who are seeing the first time our channel, please subscribe and share it to your friends. Those are preparing for the SGT or school assistant biology. Okay, very most important bits and points we are going to be discussed from the cell, the basic unit of life. Okay, see the cell basic unit of life. This is the second chapter. The first chapter is what is science. What is science already with one each and every point I have been explained, so you can visit the channel and see what is science. Then second chapter with every point with maximum covering, so we are going to discuss. Let us see the first one. The microbial world is observed by, so here microbial world, the microbial world is observed by three scientists. The major scientists who are playing important role in observing the microbial world. The first three scientists were, if you can see here, first one, what Athanasius Kircher and John Swaminathan and Anthony van Leeuwenhoek. These are the three scientists. See, Athanasius Kircher, John Swaminathan, Anthony van Leeuwenhoek. These are the three scientists. First one, Athanasius Kircher, John Swaminathan and Anthony van Leeuwenhoek. So how can we remember? First, if you write the J in middle, then both the sides you can write A. See, first one is Athanasius Kircher, last one is Anthony van Leeuwenhoek. So A plus A square, A plus J plus A. So any one code you can create and remember like this: Athanasius Kircher, John Swaminathan and Anthony van Leeuwenhoek. So like this we can create one code and remember these three scientists were very important. So in a examination he will ask you, ఈ క్రింది వాణిలో మైక్రోబియల్ వరల్డ్ని అబ్జర్వ్ చేసిన వాళ్ళలో లేని వారు అని అడుగుతారు. మ్యాక్సిమం ఉన్న వాళ్ళు దాదాపు అడుగుతారు. సో విచ్ ఆఫ్ ది సైంటిస్ట్ ఈజ్ నాట్ రిలేటెడ్ టు అబ్జర్వింగ్ ద మైక్రోబియల్ వరల్డ్? Suppose options may be Athanasius Kircher, John Swaminathan, Anthony van Leeuwenhoek and Louis Pasteur. So he may give the option like that. So the Louis Pasteur is not related to the observing the microbial world regarding this lesson. So very most important point, remember these three scientists: Athanasius Kircher, John Swaminathan and Anthony van Leeuwenhoek. These are the three scientists. Next, the first non-living cell is discovered by. Then after we are going to study about the discovery of non-living cell. Who discovered the non-living cell? Non-living cell is discovered by Robert Hooke in the year of 1665. Very most important point. See, non-living cell by the Robert Hooke. Non-living cell by the Robert Hooke in the year of 1665. Year is very important so that we can identify the living cell and non-living cell. Non-living cell is 1665, then living cell is 1674. So we can differentiate by remembering these dates. So sometimes we can also remember the years. So year 1665, Robert Hooke discovered the non-living cell. How he discovered the non-living cell? Just he took the oak tree plant bark and kept under the microscope. When he observed this non-living cell bark, he has observed some little rooms which are similar to the honeycomb hive. When we observe the honeycomb hive, there will be some small rooms or little rooms we can observe. అయితే ఈ హానీకోమ్ హైవ్లో ఎలాగైతే చిన్న రూమ్ల గదుల లాంటి నిర్మాణాలు ఉన్నాయో, అలాగే ఈ ఓక్ ట్రీ ప్లాంట్ బార్క్లో కూడా కొన్ని లిటిల్ రూమ్స్ అనేవి కనబడినవి. ఆ లిటిల్ రూమ్ ప్రతి దాన్ని ఏమన్నాడంటే ఒక సెల్గా అభివర్ణించండు. The every little room in the oak tree plant bark, that every little room is called as a cell. Generally cell is a Latin word here. When we observe the cell, see, the cell is a Latin word. Okay, the cell is a Latin word which means a little room. Very important. Cell is a Latin word. Sometimes he may ask you, what is the cell derived from Latin word, which means, okay, little room. Okay, very most important. Cell, it is a Latin word. Okay, the cell, it is a Latin word which means a little room. Next, this is all about the what discovery of non-living cell. So discovery of non-living cell by a question. Who discovered the non-living cell? And what? Robert Hooke in the year of 1665. How he discovered the non-living cell? He took the oak tree plant bark of this one layer. Oak tree plant bark, one of the layer is kept under the microscope, and he observed. When he observed under the microscope, the little rooms, small little rooms he had seen, like which are similar to the honeycomb hive rooms. So this every small little room he called as a cell. The cell is a Latin word which means little room. So this is about discovery of non-living cell. Next, living cell. We shall see about the living cell. What is living cell? Who discovered the living cell? First time the living cell is discovered by Antony van Leeuwenhoek. See, Antony van Leeuwenhoek discovered the living cell. Generally, Antony van Leeuwenhoek is not a scientist, but he is a merchant who sells the cloths. Because of his passion, he has made one single lens microscope. With this single lens microscope, he had observed the thickness of the cloth, and as well as once he observed the pond water. Basically, he was not a scientist, but he was one of the merchant. But because of his passionate to observe the pond water, he had found that there were some living creatures in the pond water moving. The living creatures moving in the pond water, these living creatures were specifically mentioned. We should remember they are called bacteria, yeast, protozoans. These three are specifically mentioned in the textbook, so we should remember. Antony van Leeuwenhoek identified which of these living organisms in the pond water. He will ask you the question. What? Antony van Leeuwenhoek క్రింది వాటిలో ఏ లివింగ్ ఆర్గానిజమ్స్ ని అబ్జర్వ్ చేసిండు అంటే ఫస్ట్ వన్ ఇస్ బ్యాక్టీరియా, ప్రోటోజువాన్, ఈస్ట్. దీస్ త్రీ ఆర్ వెరీ ఇంపార్టెంట్. వీటితో పాటుగా ఇంకా ఆర్బీసీని కూడా చూస్తాడు ఆయన, హ్యూమన్ రెడ్ బ్లడ్ సెల్స్ని కూడా చూస్తాడు. సో దీస్ 4 నేమ్స్ యూ కెన్ రిమెంబర్. ఓకే, దట్ ఈస్ అబౌట్ Antony van Leeuwenhoek. Question: How Antony van Leeuwenhoek observed the living creatures? He observed the living creatures by taking some pond water. That pond water, when he observed under the microscope, some living organisms are moving. These living organisms he observed as bacteria, yeast and protozoan. Okay, then also he also observed some RBC. So this is very important. This living organisms he called as what? Bacteria, yeast and protozoan. ఈ లివింగ్ ఆర్గానిజమ్స్ ని ఏమని పిలిచిందంటే animalcules అని పిలిచిండు in 1678. very important. In 1674 he discovered the living cells like bacteria, yeast, protozoans. But at the same time the bacteria, yeast cell, protozoans వీటన్నింటినీ కలిపి ఆయన ఏమని పిలిచిందంటే animalcules అన్నాడు. This animalcules, this word he has given in the year of 1678. This is 1674 discovery, and animalcules name is given in the 1678. So very important. So this is about the discovery of non-living cells. Discovery of living cells. Non-living cell by the Robert Hooke, then living cells by the Antony van Leeuwenhoek. Next one, if we observe the cells, suppose we can divide the living organisms, or we can divide the living cells into two. One is plant cell, another one is animal cell. Let us see the plant cell here. The plant cell is generally, if we observe the plant cell, like in the textbook there is an activity that is what, take a mastic and soak it in the water for a half an hour, then after you can peel it out, one small peel, keep it under the microscope and observe, so that we can see small cells, this type of cells. Okay, this type of cells we can observe. Whereas if we take onion peel cells, take an onion, break it in the middle, so take it out a thin layer of onion peel and keep it under the microscope and observe. Same you can see this type of cells. So these cells are, what is the structure of these cells, or what is the shape of these cells? If you observe these cells here, the cells are in rectangle in shape. If we observe these cells, we can observe the shape of the cells were rectangle in shape. In these cells, the cell's boundary, if we observe the boundary of the cell is very thick in nature. Why this boundary of the cell is very thick in nature? Because of here there is a specific character which is present in the plant cell, that is called cell wall is present in the plant cell. The very important character, the cell wall is present in the plant cell. This is an extra membrane we can observe only in the plant cell. So generally cells consist of cell membrane. Okay, cell membrane is common in both the plant cell and animal cell. Cell membrane is common, but here the extra character is cell wall. The cell wall is only present in the plant cell. Because of the presence of cell wall, the cell boundary, the cell is very thick in nature. This is the basic character, remember, about cell wall. Then we will study something more in the 9th class, what is the function of cell wall. But here just you can remember why the plant cells were very thick. Why the plant cells are very thick? Because the cell wall, this is an extra layer which is present over the cell of plant cell. That's why the cells are very thick. And next, cell membrane is also present. Below the cell wall there is also cell membrane. Cell membrane is present in the both plant cell and animal cell. We can observe. Extra layer in the plant cell is cell wall. And next one, if you observe the cell inside, here there is a nucleus. See, the nucleus is just beside it in the plant cell. The nucleus is beside it. And the cytoplasm is heterogeneous cytoplasm. Very important character. Cytoplasm is heterogeneous in character. Heterogeneous character. So cytoplasm character is heterogeneous. That means if we can observe the cell is rectangle in shape. Then nucleus is just beside it, because most of the plant cell is occupied by the vacuole, the cell organelle. That's why the nucleus is not in the center, beside it. And cell membrane is common in the plant cell and animal cell, but the extra layer is cell wall is. Next, cytoplasm is present. This cytoplasm character is very important, heterogeneous. Underline this word, heterogeneous. The cytoplasm is heterogeneous in nature. Next, nucleus. We shall study about the nucleus present in the cell. Here we have discussed now the nucleus is present in the plant cell, whereas nucleus is also present in the animal cell. What type of nucleus it is? Nucleus is here besiding. In plant cell there is besided nucleus. This consists of, okay, here we can see the nucleus is besided, because the most of the plant cell is occupied by the, suppose example, see, the most of the plant cell is occupied by the vacuole. This is one of the cell organelle. Because of the presence of vacuole, more it is occupied, so the nucleus is besided in the plant cell. So the nucleus, if we can see, first time the nucleus is observed by the Felis Fontana in the where epithelial cells. Generally epithelial cells are present in the animals. Epithelial tissue. See, our skin is one of the epithelial tissue. So like this, in epithelial cells, first time the cell is observed by the Felis Fontana. Very most important. Cell is discovered by Robert Brown, but cell is first time observed by the Felis Fontana. Then after observing the nucleus in epithelial cells లో Felis Fontana nucleus ని అబ్జర్వ్ చేసిన 180 years తర్వాత, after 180 years, very important, that's why I have written here in inverted commas. See, after 180 years Felis Fontana అబ్జర్వ్ చేసిన న్యూక్లియస్ ని, అంటే ఎపిథెలియల్ సెల్స్ లో Felis Fontana న్యూక్లియస్ ని అబ్జర్వ్ చేసిన 180 years తర్వాత రాబర్ట్ బ్రౌన్ డిస్కవర్డ్ ద న్యూక్లియస్ ఇన్ ఆర్కిడ్ సెల్స్. ఈయన ప్లాంట్ సెల్స్ లో కనుక్కుంటాడు. రాబర్ట్ బ్రౌన్ డిస్కవర్డ్ ద న్యూక్లియస్ ఇన్ ఆర్కిడ్ సెల్స్. అండ్ ఈయన ఏం చెప్తాడంటే, దిస్ న్యూక్లియస్ ఈజ్ అన్ ఇంటగ్రల్ పార్ట్ ఆఫ్ ది సెల్ అని చెప్తాడు. న్యూక్లియస్ ఈజ్ one of the most important part of the cell which controls all the cell organelles. Okay, nucleus which is also an integral part of the cell, ani ila clear information he has given. That's why the nucleus is discovered by Robert Brown. We will say Robert Brown discovered the nucleus, and he also said that nucleus is an integral part of the cell. That's why the nucleus discovery, that credit is goes to, goes to Robert Brown in the year of 1831. So this year also remember 1831. Okay, nucleus in the year of 1831. Robert Brown discovered the nucleus. Then we will study more about the nucleus in the 9th class. Generally, nucleus is one of the cytoblast, because the new cells are produced from the nucleus. We can also call them as cytoblast, who is said by the Schleider. And also next one more, nucleus which consists of genetic material. That genetic material consists of genes, chromosomes carry some genes are responsible for characters, so that the characters can be transferred from one generation to another through the nucleus. Or nucleus consists of chromosomes, or chromosomes consist of genes which can transfer the characters from one generation to another. So this is the information that we will something more we will study in the 9th class. But in the 8th class, just we can remember the nucleus. First time nucleus is observed by the Felis Fontana in the epithelial cells, but after 180 years the Robert Brown discovered the nucleus. This nucleus, it is said as an integral part of the cell, and also which controls all the cell organelles. So that's why the nucleus is called as cell controlled room, and you'll have clear information each and about nucleus by the Robert Brown in the year of 1831. So this is all about the nucleus. And one more important thing here, diversity in cells. Next one, if we see about diversity in cells, ఇక డైవర్సిటీ ఇన్ సెల్స్ కనుక చూస్తే, బిఫోర్ దట్ జస్ట్ వీ విల్ సీ ద animal cell. Suppose animal cell, if we can observe, animal cell is circular in shape or irregular in shape. See, the animal cell is circular or irregular in shape. The animal cell which consists of nucleus in the center. See, we can observe the nucleus in animal cell is center, but nucleus in the plant cell is beside it. Then the cell membrane in animal cell is present here also cell membrane is present. That means cell membrane is common in both the plant cell and animal cell. See, this is the cell membrane. The outer membrane of the animal cell is cell membrane, whereas here outer membrane of the plant cell is cell wall. Below the cell wall there is cell membrane, so that cell membrane is common. Next, here the cytoplasm is heterogeneous, here is also cytoplasm is heterogeneous, but the common character is cell membrane is common in both the plant cell and animal cell. Okay, then here plant cell is rectangle in shape, whereas animal cell is circular in shape. We can compare like this: the cell wall is only present in the plant cell, but cell wall is absent in the animal cell. Okay, this type of some common characters, so we can observe differences and some common characters we can observe in between the animal cell and plant cell. And next we shall see diversity in cells. Diversity means differences. We shall see the differences in the cells. See, diversity in cells. What is the diversity? The diversity means differences. If we see the differences, here we can observe the TS of Stridot system. In the textbook we can see TS of Stridot system. TS of Stridot system lo, so this is the first layer. The first layer is, if we can observe, these are A group A cells. Ikkada outer layer of the cell, that A group cell or group A cells, which are for the protection and also give shape to the cell. That means the dermal tissue cells, okay, which give the protection and also give the shape to the plant. Generally shape of the cell is given by the plasma membrane here, but A group cells, generally plasma membrane is also an outer layer of the cell. Here is also we can see the outer membrane, that is dermal tissue. The dermal tissue cells are which are used for the protection and also give shape. This is the A group cells. So this is one type of cells which are present in the TS of stem. Then B group of cells in green color. In next layer, see the B group of cells. These B group of cells are green in color. Generally, if the cells are green in color, we can understand which perform the photosynthesis. So they are nothing but chloroplast. So chloroplast, either it is maybe present in the leaf or stem or root. So wherever there is the chloroplast it can perform the photosynthesis. It traps the solar energy and associate with carbon dioxide, water, sunlight, and prepare the food by itself. So it is called as, that's why we are calling plants are autotrophs. So the B group of cells for photosynthesis, whereas C C group of cells for the transportation of water and food materials. So this type of tissue generally we call it as vascular tissue. Vascular means the vascular tissue which consists of xylem and phloem. We will study in the further classes. Xylem generally transport the water, whereas phloem can transport the food materials. So the third group of cells, see the third group of cells are generally, see this third group of cells are for the transportation of water and food. Here for transportation of water, here we can observe the xylem, and transportation of food material by the phloem. And last D group of cells, this D group of cells generally it is with matured stem. This is grown as a one of the wood. This is matured stem. So like this, only the TS of stem. The TS of stem means the stem is only one. If we cut the transverse section of the stem, if we can observe, the stem is, the plant is only one, but we can observe the 4 different types of cells here. A group of cells for the specially protection, B group of cells for the especially for preparation of food material, C group of cells for the transportation of water and food, whereas D group of cells are different. So in this we can observe 4 different types of cells in a single stem. That's why we can observe there is diversity in the plants. And whereas diversity in animal cells, we can also observe the diversity in animal cells. See diversity here in our body, in the human body, if we can observe, there is a smooth muscle cell. Smooth muscle cell, this is called a non-striated muscle. In this muscle there is no striations. And also bone cell, we can also observe the bone cell. We can also observe the nerve cell. In the diagrams we can observe, see here, this is the smooth cell, bone cell, nerve cell and striated muscle cell. In striated muscle cell there is striations. So this is striated muscle cell, this is smooth muscle cell, this is bone cell, this is nerve cell and RBC. So in WBC again different types. See here eosinophil, basophil, neutrophil. See eosinophil, in eosinophil there is bridge shaped nucleus. WBC generally there are 2 types, granulocytes, agranulocytes. Granulocytes are classified into 3 types, agranulocytes are classified into 2 types. Granulocytes 3 types, what? Eosinophil, basophil, neutrophil. See eosinophil, generally eosinophils, if we can observe eosinophil, there is granules inside the cytoplasm, and in eosinophil there is a bridge shaped nucleus. See there is a bridge shaped nucleus. This bridge shaped nucleus if we can observe, that is called eosinophil. Eosinophils are increased during the allergic condition, whereas basophil, S-shaped nucleus is observed in the basophil. These basophils are generally used for the healing of wounds or curing purpose. And in neutrophils, many lobe nucleus, see this many lobe nucleus is observed in the neutrophils. When we compare these basophil, eosinophil, neutrophils, the neutrophils numbers were very, most of the number of WBC are neutrophils. They are called as microscopic policemen of the body. They protect us from the microorganisms or from the infections. That's why we are calling the neutrophil is called a microscopic policeman of the body. And next one is the agranulocytes. Agranulocytes means the granules are absent in this. These are 2 types. One is monocytes, another one is lymphocytes. In monocytes we can observe the kidney shaped nucleus. Especially here nucleus is prominent to observe the WBC. So monocytes nucleus is kidney shaped nucleus. So this is for the, they are also called the phagocytes. That means if there is any microorganism or foreign bodies enters into the cell or into the body, they phagocytes. That means they engulf this material and clean the body. That's why we are calling, this is called monocytes. And lymphocytes generally, these lymphocytes are offering the immunity power to the body. So immunity, that means protection against the diseases or resistance power of the body comes from the lymphocytes. So like this WBC 2 types, agranulocytes, granulocytes. Okay, agranulocytes 2 types, monocytes, lymphocytes, whereas granulocytes 3 types, eosinophils, basophil, neutrophils. If we observe all these cells, we can understand that the cells present in the human body are different. That shows about diversity in nature. So even the body is made up of with a number of cells. Each cell is specifically different to perform the its own function. So that bone cell is there, nerve cell is there, smooth muscle cell, non-striated muscle cell, striated muscle cell, red blood cells. See the RBC is generally biconcave in shape without nucleus, whereas WBC is different cells. So by understanding this all, The cells are different or diversity in their nature. And next one, suppose if we observe the animal cell of amoeba, it doesn't have any shape because the pseudopodiums are present. Pseudopodium means, see, suppose if it is an amoeba, here is the amoeba, these are the pseudopodiums. These pseudopodiums are generally not in constant manner, they always change. Pseudo means false, podium means field. So pseudopodium, that means false feet is present. False feet means the feet is continuously changing, they are not in the true manner, they are changing continuously in their lifestyle. So we are calling it doesn't have any shape. If the pseudopodiums are changed, then automatically the living body shape is also changed. That's why amoeba doesn't have any shape. So amoeba doesn't have any shape. And also one more important thing, the largest cell in the animal kingdom, the largest cell in the animal kingdom is ostrich egg. The largest cell in the animal kingdom is ostrich egg. Its length is, its diameter or its measurement is 17 into 18 centimeter. Okay, the largest cell is ostrich egg, it is 17 into 18 centimeter, whereas smallest cell is mycoplasma, it's 0.1 to 0.5 microns. Its length is 0.1 to 0.5 micron. This is called a bacteria. Mycoplasma is a one type of bacteria generally which is responsible for causing some diseases like pneumonia in cattle or in living organisms. Whereas kidney or liver cell is generally 20 to 30 microns. Very most important to be remembered, 20 to 30 microns. Kidney and liver cells are 20 to 30 microns. And nerve cell is 90 to 100 centimeters. The what nerve cell? Centimeter. Sometimes we may confuse either centimeter or millimeter or micrometer. So be careful. Nerve cell is 90 to 100 centimeter, whereas kidney and liver cell is 20 to 30 microns. Okay, and smallest cell mycoplasma is 0.1 to 0.5 microns, and whereas largest cell is ostrich egg, and ostrich egg it is 17 into 18 centimeter. And next one, here we can also see some measurements regarding the meter, centimeter, millimeter, nanometer. Okay, 1 meter is equal to 100 centimeters, 1 centimeter is equal to 10 millimeters, 1 millimeter is equal to 1000 micrometer, 1 micrometer is equal to 1000 nanometers. So sometimes we may ignore this, but this part also very important. Okay, many times he will ask the question from this also. Each and every point I have been covered regarding the second chapter, the cell, the basic unit of life. Once again I summarize what we have been studied in the second chapter, the cell, basic unit of life. Here what type of points to be remembered mainly. First, the cell is discovered, non-living cell is discovered by Robert Hooke in the year of 1665 from the oak tree plant bark. That is very important. The cell is a Latin word which means little room. Then, and one more important thing, the living cell is discovered by Antony van Leeuwenhoek in the year of 1674. Then Antony van Leeuwenhoek, he is not a scientist, he is a merchant. Because of discovery of one single lens microscope, this single lens microscope is magnified 300 times more. This is also very important. Antony van Leeuwenhoek తయారు చేసిన సింగిల్ లెన్స్ మైక్రోస్కోప్ ఎన్ని రేట్లు ఎక్కువ చేసి చూపెడుతుంది అంటాడు. 300 times more, or it can magnify and show. So that is very important. And next one, this living organisms who discovered the under the water is bacteria, protozoan, yeast. Yeast is a single celled fungi. Bacteria we know that. And next one, protozoan. Proto means first, zoa means animal. So like this, protozoan, bacteria, yeast cells were discovered by Antony van Leeuwenhoek, and also he observed the RBC. And another important point, nucleus is discovered by Robert Brown in the year of 1831. But before the Robert Brown, Felix Fontana observed the nucleus in epithelial cells. After 180 years, Robert Brown, he said that nucleus is an integral part of the cell. So that in 1831, the nucleus was discovered. And next one is diversity in cells, diversity in animal cells. Then plant cell, animal cell differences. Generally, plant cell rectangle in shape, whereas animal cell is circular in shape. Plant cell is nucleus beside it, whereas animal cell nucleus is center of the cell. When plant cell, it has cell wall, but animal cell it doesn't have cell wall. These common points, these are the very most important points, remember. And next one, the smallest cell is mycoplasma, 0.1 to 0.5 microns. Largest cell is ostrich egg, 17 into 18 centimeters. Then kidney liver cell, 20 to 30 microns. Nerve cell is 90 to 100 centimeters. So these are the very most important points that we have been covered in the second chapter, that cell, the basic unit of life. For more updates, please subscribe and continue the